Hello everyone, I'm happy to be here for this virtual conference. My name is McKinnon, I'm from Brigham Young University, and I'd like to present to you a comprehensive analysis of the phylogenetic signal in ramp sequences in 211 vertebrates. So first off, what exactly is a ramp sequence? Well, a few years ago, researchers discovered ramp sequences as a means of regulating gene expression rates. The way that it does that is that many times in genes, at the beginning of the sequence, you'll find rare codons that aren't used very often in the genome that cluster here at the beginning. This is important because since these codons are rare, there are less tRNA molecules in the cell available to translate them. This means that as the ribosome is translating, it will take longer at the beginning of the gene as it waits for the tRNA molecules to associate. This is especially important in highly expressed genes because it means that as multiple ribosomes attach to the same gene to translate it, then they'll move slowly throughout this beginning area and it will reduce any possible ribosomal collisions, and it will also reduce translational errors. And it's been previously estimated that ramp sequences exist in approximately 15% of genes. So they're actually a pretty prevalent way of regulating gene expression that is encoded right into the DNA sequence. A couple years ago, researchers in my lab developed an algorithm called XRAMP, which is able to identify a ramp sequence in a single gene. It does that in 3 steps. First, it calculates the relative codon efficiencies using codon frequencies. So that means that codons that are used more frequently in the genome are estimated to be translated more quickly than codons that are used very rarely. It then calculates the ribosomal speed at each location in the gene by averaging the mean codon efficiency of a 9-codon sliding window. And we use a 9-codon sliding window as the estimate for the length of the ribosome. And this step results in an estimate for gene translation throughout the entire sequence. The third step is to locate any outlier region that might exist at the beginning of the gene where translation happens more slowly. In this particular project, we built off of this previous research and we asked the questions, are ramp sequences phylogenetically conserved? And can ramp sequences be used as a novel phylogenetic character? Addressing that first question, our methodology was to first download species FASTA files from NCBI database, and we downloaded all of the vertebrate species that were available at the time. And we then used XRAMP to find any ramp sequences that might exist in their genome. Our third step was to encode each ramp sequence in each orthologous gene of the species as ramp sequence present or ramp sequence absent. And we encoded that in a binary matrix, which we would then use for our phylogenetic inference. Our fourth step was to filter the ramp sequences. So we removed any ramp sequences that were only found in a few species so that we could limit the effect of missing data. Our fifth step was to recover the phylogenies using parsimony and maximum likelihood. And sixth step was to compare to reference phylogeny to see how well a phylogeny that was recovered using only ramp sequences compares to phylogenies that are more established, like the open tree of life. Our ramp sequence phylogenies resulted in these phylogenies here for mammals and non-mammalian vertebrates. But as you can see, we needed a way to quantify whether or not these phylogenies are any good. So we used the branch percent identity with the open tree of life. The open tree of life is based on a number of different studies, and it represents a good estimate of what we currently know to be species relationships to be. And we found that our ramps phylogenies with parsimony and maximum likelihood were able to recover approximately 70% of relationships. And we compared this to other genomic attributes like codon inversion, amino acid inversion, and other word-based methods. And we determined that This percent identity is very comparable with many other commonly used phylogenetic algorithms. Our next question was, can ramp sequences be used as a novel phylogenetic character? And we addressed this by mapping each ramp sequence in each orthologous gene to the open tree of life, and then we counted the number of parallelisms and reversals that occurred for each ramp sequence. This was quantified using the retention index of ramp sequences, and just to simplify, a retention index of 1 means that a character is completely homologous, whereas a retention index of 0 means that the character has no phylogenetic information. When we plotted the retention indexes of all of our ramp sequences, we found that many of them do contain phylogenetic information, although some of them also appear to be homoplagious, which you can see by these zeros here, for both mammals and non-mammalian vertebrates. And we wanted to know if these retention indexes were any good, so we compared that to 1,000 random permutations, seen here. What we did was we took the open tree of life and we shuffled the species in it 1,000 times, and we went through this same process of calculating the retention index over and over again. And we found that our random permutations were all very low as for their mean retention index, whereas our observed retention index was way higher, as you can see by the dotted line here to the right. And the same pattern can be seen in non-mammalian ramps. And this results in an empirical p-value of 0, showing that ramp sequences do contain phylogenetic information. Just to conclude, we learned in this project that the presence or absence of a ramp sequence in many orthologs contains enough phylogenetic information to recover many species relationships in the open tree of life. And we also learned that the retention index is higher than would be expected due to random chance. Although some ramp sequences are homoplagious, if you look at the overall usage of ramp sequences, we see a statistically significant level of homology as compared to the open tree of life. And we suggest that ramp sequences should be used as a novel characteristic in future phylogenetic studies. I'd like to briefly thank my mentors in the Fulton Supercomputing Lab for supporting the project, and I'd be happy to take any questions. Thank you.